Welcome to the recovery training for Paxlovid. This slide shows the current recovery treatment comparisons for adults hospitalised in the UK with COVID, excluding the comparisons for flu. Paxlovid is an oral antiviral drug being introduced into recovery at the end of March 2022. It will become one of 3 antivirals in the trial, along with sotrovimab and molnupiravir. As recovery has a factorial design, the 5 comparisons shown here are all independent, meaning that patients can be entered into all of those they're eligible for, in the opinion of their clinical team. Each is an independent coin toss, so patients can be allocated to any combination of the treatments that they're eligible for, including none or all simultaneously. Paxlovid is the brand name for a combination of 2 drugs, nirmatrelvir and ritonavir, that are packaged together, although not actually co-formulated in the same pills. Nirmatrelvir, which until recently was usually referred to by its compound number, is an inhibitor of the main SARS-CoV-2 protease. This enzyme cleaves the viral polyprotein following translation, and by inhibiting it, nirmatrelvir prevents viral replication. It's the first approved small molecule drug specifically developed for COVID-19. Ritonavir has no antiviral activity itself, but is included to inhibit metabolism of nirmatrelvir, resulting in higher blood concentrations. This is the same as its use with HIV protease inhibitors like lopinavir, which was one of the first drugs tested in recovery. Unlike the spike protein, the protease is relatively conserved, so Paxlovid retains activity against Omicron and will hopefully be active against future variants too. Because the mechanism of action of Paxlovid, molnupiravir and sotrovimab are distinct, they could have additive effects when given together, which is why the randomization system allows patients to be allocated them alone or in combination. Paxlovid is approved for use in early COVID on the basis of the EPIC-HR trial, but there's no data on the effect of the drug in hospitalised patients. In EPIC-HR, 2,000 patients with COVID symptoms starting in the past 5 days were given Paxlovid or placebo. Hospitalisation or death occurred in 8 patients in the Paxlovid group versus 66 in the placebo group, a reduction of nearly 90%. There were no major adverse effects of Paxlovid identified, but there was a definite excess of altered taste, which occurred in 6% of patients. And 3% of patients on Paxlovid had diarrhea, which was slightly higher than placebo. However, overall discontinuation of the drug because of adverse events was higher in the placebo group. There are several contraindications to enrolment in the Paxlovid comparison. Patients under 18 and those with severe liver or renal impairment are excluded. And there are several significant drug interactions that I'll cover on the next slide, which may be a contraindication if the interacting drug can't safely be held whilst on Paxlovid. The tablets can't be crushed and there's no IV preparation, so patients must be able to swallow. And if a patient's already received Paxlovid during their current illness, then they're ineligible. Women in the first trimester of pregnancy are excluded, but those in the second and third trimesters, or who are breastfeeding, are potentially eligible. Inclusion of these women needs careful discussion, as although there have been no concerns in animal models, nirmatrelvir is a new drug and there's no human pregnancy safety data. There's a new requirement in the latest protocol version to discuss the potential inclusion of pregnant women to any trial arm with an obstetrician or obstetric physician. More information on this is in the site team's page of the recovery website and in the protocol appendix. Because ritonavir, like clarithromycin, is a potent inhibitor of cytochrome P453A, it can increase the level of other drugs that are also substrates, and some of these interactions are dangerous, for example with specific statins, anticoagulants, antiarrhythmics, antipsychotics, benzodiazepines or immunosuppressants. We suggest using the University of Liverpool COVID-19 drug interaction checker, shown here, which now includes Paxlovid. This allows potential interactions to be easily identified and has a summary table of interacting drugs, as well as a specific flowchart for Paxlovid. If a particular drug can't be found in the checker, then you can refer to the protocol list of contraindicated drugs in the appendix, or to the SPC. Dexamethasone levels are increased by ritonavir, so patients can't also be randomised to the high-dose dexamethasone comparison, but this shouldn't have an effect on normal-dose dexamethasone for COVID. Other recovery drugs and standard COVID treatments are unaffected, including tocilizumab, baricitinib, remdesivir, ronapreve, sotrovimab, molnupiravir, and empagliflozin. The dose is Paxlovid 300-100 twice daily for 5 days, which are 2 150 mg of tablets of nirmatrelvir and 100 mg tablets of ritonavir. In patients with moderate renal impairment with an eGFR of 30 to 59, this dose should be reduced to 1 nirmatrelvir tablet and 1 ritonavir tablet twice daily. In both cases, the course should be completed at home if the participant's discharged before it's finished. Because time to discharge is a trial outcome and there's no placebo group, please try to ensure that provision of drugs to take home doesn't delay discharge, otherwise it could potentially bias the result for this outcome. If possible, dispensing the whole course to be kept on the ward with the patient may minimise this risk. Another effect of ritonavir is to affect the reliability of the combined pill, so women of childbearing potential should use an effective alternative for at least one cycle after stopping, which is mentioned in the participant information sheet. As with other antiviral comparisons, we think that biological sampling may be crucial to identify particular patients that benefit from treatment, as was the case for ronapreve. This slide shows a sampling plan, which is the same as for the sotrovimab and molnupiravir comparisons. 2 different sample types are being collected. Firstly, a serum sample, which will be used to look for SARS-CoV-2 antibodies and for viral antigen. And secondly, nose swabs, which we'll use to look at the effect of the antiviral drugs on the rate of viral clearance, and also to look at the presence of mutations that might confer resistance to the antivirals. At each time point, a single swab is used to swab both nostrils. Both samples are taken at baseline, which is day 1, and these must be taken after consent's been given, but before randomization. Swabs will also be taken at day 3 and day 5. Sites should have sample kits for serum and nose swabs, and these include a return envelope so they can be posted in the hospital internal mail. We aim to keep sites stocked up with these, but please email us if you're running low. Instructions for taking samples are in each kit and can also be found on the recovery website on the site teams page. A new development in the latest protocol version, starting April 2022, is that if patients are discharged before day 5, they should be asked to take nose swabs themselves after discharge if possible, using the usual recovery kits. Samples are taken on the same days as they would be in hospital, and then patients can post a kit themselves in a postbox, so this won't need the involvement of the local team. Instructions for self-swabbing are on the recovery website, so please read these and print a copy for the patients if needed. It may be helpful to leave pre-labelled day 3 and day 5 swab kits with patients that could be discharged at short notice, even if this means that they occasionally get lost. Self-swabbing will apply to patients in all the antiviral comparisons, not just Paxlovid. And as before, it's really important that samples are taken not only from people allocated the antiviral treatments, but also those allocated to usual care in the antiviral comparisons. In other words, we need samples from everyone that might have been allocated an antiviral at randomization, whether or not they actually were. Please email us at the main trial address if you have any questions. Thanks for listening.